Welcome to EduRev.
Welcome students. I'm Clarice Pereira Colasso and I have been training NEET medical aspirants in botany and zoology as well as NCERT based board exams for the past 8 years. We have already completed the pre-fertilization structures in the previous videos, the structures being the structure of the androecium and the structure of the gynoecium. We have also completed a number of pre-fertilization events, and those would include microsporogenesis, so the formation of the pollen grain, megasporogenesis, so that's the formation of the megaspore and its development into the embryo sac. We have also studied pollination and the various types of pollination. So in this video, we will be completing the remaining pre-fertilization events, those being pollen tube germination. So what is the pollen and the pistil interaction going to be like? Artificial hybridization, this is required for crop breeding programs. And finally, we'll go into the main event, that is the fertilization event. So to start off with pollen grain germination, we must understand that pollen, the moment pollination takes place, pollen grain have fallen onto the stigma and a pollen pistil interaction begins to occur. However, we must remember for pollen pistil interaction to occur in a proper fashion, the type of pollen grain falling onto the stigma must be viable pollen grain. And viability of pollen grain is governed by two factors, those being temperature and the relative humidity of the area. Okay, so we need to remember these 2 points. Now, these 2 points are very important with regards to your NEET exam. Now, the pistil consists of 3 different parts as we have already studied. So that is going to be a stigma, the style and the ovary. Now, considering it is of 3 different parts, the pollen grain must have an interaction with all these 3 parts. So let's try to understand what are those interactions. Firstly, pollen has an interaction with the stigma. So you can very clearly see the moment pollination takes place, pollen grain has fallen onto stigma, pollen tube germination begins. Now, what is really pollen tube germination? If you remember, the pollen grain consists of 2 cells. It can either be 2-cell stage or 3-cell stage. Let's assume we're talking about a 2-celled pollen grain because normally speaking, anther dehiscence and pollination requires or has these particular 2-celled pollen grains moving about from the anther to the stigma. So assuming that this 2-celled pollen grain falls onto the stigma, the vegetative cell is also called as the tubal cell. So this particular vegetative cell begins to start growing. It begins to start elongating. And if you remember, it starts moving out through what we call as the germ pore of the exine. This is the only part of the exine that does not have the presence of sporopollenin, which is considered as the most resistant organic material. Okay. Also, remember that the male gametes in the 3-cell stage, or for that matter, even right now, the generative cell of the 2-cell stage, they're not separated from the vegetative cell. However, they lie in the cytoplasm of the vegetative cell. So the moment the vegetative cell begins to elongate, the generative cell or the male gametes will flow along with that particular vegetative cell cytoplasm. Now, if you look at the figure in front of you, you will see that some of the pollen grain that have fallen onto the stigma have resulted in this particular pollen tube formation. However, there are 2 very clearly over there that have not resulted in pollen tube formation. Assuming that all of these were viable pollen grain, the question is, why did these 2 not really form a pollen tube? And the probable answer is because of a concept called as self-incompatibility. So if you remember, when we studied about outbreeding devices, we said cross-pollination is preferred. So we would prevent self-pollination. How do you prevent self-pollination? If self-pollen does fall onto the stigma, do not allow it to form the pollen tube. So that is the concept of self-incompatibility. So here we're making an assumption when we say most likely those that have formed pollen tubes are going to be cross-pollen and not self-pollen. Now, coming to the second part of the interaction, that is the interaction between pollen and the style, so the elongated portion. Now, the question is, how does the pollen tube know that it needs to move in that particular direction? So there is a chemotropic attraction that is causing it to move downward. There's a gradient that helps to move downward, and this gradient is of calcium, boron and inositol. Okay, so we need to remember this, very important for your NEET exam. Now, the last part, the pollen tube must have some sort of an association or some sort of a relation with the ovary. So as it begins to approach the ovary, it needs to ultimately interact with the egg cell, and it needs to interact with the pollen nuclei. We'll very soon understand why. And if you remember, when we spoke about the embryo sac, we have the presence of 2 synergids having filiform apparatus in each of them, so this finger-like projections. And we said the purpose of this filiform apparatus is to guide the pollen tube upward towards the egg cell. Okay, so in other words, the elongated vegetative cell is going to move into that particular direction, which means the male gametes also will move. Now, how does the filiform apparatus really help in the movement of the pollen tube? How does it take it upward towards the egg cell? Now, the answer is as simple as this. By allowing these finger-like projections, first of all, to help it move, okay, and secondly, by also degenerating on its own. So you will see that it will begin to degenerate after some time, okay. So as it begins to degenerate, it will release these male gametes close to the egg cell nucleus or close to your pollen nuclei, depending. So here you can see degeneration of the syzygium that allowed pollen tube entry. One male gamete very close to the egg cell, one male gamete apparently very close to the pollen nuclei. So this is the interaction of the pollen tube with the ovary. Now we move on to the concept of artificial hybridization. So hybridization basically implies that you're bringing two different plants together, but here artificially, so artificial hybridization. So artificial hybridization is mainly a technique by which we allow pollination between two plants of our choice. So it is a crop breeding program. It is an event in the crop breeding program. Okay, so a technique that uses only desired pollen for pollination. So you're bringing the two plants of your choice together. Now, in order to conduct artificial hybridization, you have numerous number of steps, but the main steps include something called as emasculation, which means removal of the masculine part, which means in other words the stamens or the anther, and the second is something called as bagging and rebagging. So we will understand that as we move along. Now we have a small flower over here, a purple color variety of a flower, and very clearly in the figure you can see you have a petal, you've got a stigma, you've got the anther, stamen and carpel. So the moment you have the stigma and the anther together, you can easily understand that this is a bisexual flower. Now the problem in a crop breeding program is this. If I want to allow pollen of a very specific plant to fall onto the stigma of this particular plant, let's say this purple color flower, we have a problem when you have a bisexual flower. I cannot be 100% certain that the stigma will contain only my pollen of choice. It may contain my pollen of choice, but it may also contain self pollen. Okay, so there could be problems with this particular bisexual flower. Now let us assume that we want only the female part of this purple color flower, and we have another flower of the same species but a white color variety. Okay, and I want the male part of this particular flower, which implies I'm going to collect pollen from the white color flower. So let us make an assumption that we are conducting a crop breeding program and we have simply collected pollen from the white color flower and we have kept it aside. Now we're going to deal a little bit with the purple color flower. The easiest method or the easiest thing for us to do is to cut off the stamens. So emasculate the flower. If you cut off the stamens, though it was a bisexual flower, in a way you have converted that bisexual flower forcefully into a unisexual flower, so only the stigma remains. Now, the moment you emasculate a flower belonging to a plant, it is important that you cover this particular flower. Remember, this is only the female part now. So it is important that you cover this entire plant, so in a way cover it with a polythene bag probably, okay, until the stigma becomes receptive. In other words, until the gynoecium is fully matured. Now the moment the gynoecium is fully matured, you will transfer pollen from the white flower to the purple flower because that was what you wanted to do. The moment you transfer pollen from the white flower to the purple flower, we again need to rebag the purple color flower because remember the stigma is going to be receptive and there could probably be stray pollen in the air, so you want to prevent any sort of unwanted pollination. So you will conduct what we call as rebagging. However, you need to understand, pollen has now been put onto the stigma and pollination will take place, seed formation will take place. So ultimately the fruit also will form over here. So in other words, at the end of the entire process, you will find that in that polythene bag, during rebagging, you actually rebag just the purple color flower. But at the end, you will get the fruit also. Okay. So during rebagging, at the end of rebagging rather, you will actually be able to see the fruit instead of the flower in that polythene bag. Okay. So I hope you have understood the various steps of artificial hybridization, emasculation, bagging, and rebagging. Now we move into the main part of our chapter, and that is fertilization. Now fertilization in case of angiosperms is said to be double fertilization, implying there are two fertilization events that are taking place over here. So we're going to study these two fertilization events separately. Now let's try to understand why do these two fertilization events first of all come in picture. Remember, you have two male gametes. One male gamete very close to the egg cell, another male gamete very close to the polar nuclei. So both these two male gametes will result in different fusion products and therefore two different fertilizations. Let's look at it in depth. The first event of double fertilization. Now, the first event of double fertilization is called syngamy. Syngamy implies synchronized gamete fusion. Okay, so very fact that you say synchronized gamete fusion, we are talking about a haploid egg cell with a haploid male gamete. Both of them are fusing together, so you have a fusion product to result in a diploid zygote. This is but obvious when we have normal fertilization or normal syngamy. Okay, now remember that this particular zygote is a single cell, and this particular zygote must undergo mitosis in order to increase its number of cells. So in other words, in order to undergo embryogenesis, you need to have the presence of mitosis and therefore form the embryo at the end. So this is the first event of double fertilization. Now coming to the second event of double fertilization. The second event of double fertilization is called triple fusion. The very fact that we have this word triple fusion rather than the word syngamy, it indicates that probably three nuclei are being fused together. Now the question is, what are the three nuclei that are being fused together? You have the two polar nuclei and the one male gamete. Or for that matter, you could have considered one central nucleus and one male gamete. But when you consider one central nucleus, one male gamete, you don't really have three nuclei as such fusing. So an easy way to understand triple fusion is you have three haploid nuclei that will fuse together. Which are the three? The single male gamete and two polar nuclei. So triple fusion. The moment triple fusion takes place, obviously you have a triploid nucleus that will come into picture. So fusion results in the formation of a cell called as the primary endosperm cell. It is a single cell. And this particular cell contains what we call as the primary endosperm nucleus, called as the PEN. Okay? And this is a triploid nucleus. Now, this particular triploid nucleus or this particular PEC, primary endosperm cell, will also undergo mitosis like the previous zygote. Okay? But when it undergoes mitosis, it will result in the formation of an endosperm. Now we need to understand what is really the function of the endosperm. The endosperm contains a triploid nuclei, which means a whole lot of DNA. A whole lot of DNA would also imply a whole lot of RNA and therefore a whole lot of proteins, which means ultimately speaking, you have a whole lot of nutrition being produced by the endosperm. This particular nutrition will then be provided to the zygote for its development. In other words, the zygote depends on endospermic nutrition in order to carry out mitosis and therefore result in the embryo. Okay, so the zygote is actually dependent upon the endosperm. Now, let us go in through for a couple of questions. The first one. Vegetative fertilization indicates. So what is really vegetative fertilization? Now we've now come across this term vegetative fertilization. But if you remember, we spoke about a double fertilization. One double fertilization resulted in the formation of a zygote, that was syngamy. The second second event of double fertilization resulted in the endosperm or the primary endosperm cell. Now there's a difference over here. In the first case, egg cell plus sperm, or rather egg cell plus male gamete, gave rise to the zygote, which means this was generative. It generates the new generation, gives rise to the new generation. But polar nuclei plus male gamete did not give rise to a new generation. So triple fusion event can also be called as vegetative fertilization, whereas syngamy will be called as generative fertilization. So let's go through this MCQ. Vegetative fertilization indicates dash. A. Fusion of the vegetative cell of the male gamete with the nucellus cell. Now we know that that's not true. There is no fusion of male gamete with the nucellus. Next. Fusion of the second male gamete with the central cell. Now this could probably make sense because the central cell, in other words, is a two polar nucleus. Probably the answer is B, but let's go through the other MCQs, other options. Point C. Fusion of the two polar nuclei to get one central cell. Now we know this is not the correct thing. This is what happens during embryo sac formation, and D is none of these. So definitely the answer here is option B. Second one. Pollen tube grows chemotropically due to dash. Calcium mostly, magnesium mostly, boron mostly, calcium boron inositol complex. So this is very straightforward. We've already discussed it. The answer here is D, that is calcium boron and inositol complex. Next, Which of the following gives a correct chromosome number for each of these entities? Option A. Pollen grain N, XL 2N, zygote 3N, PEN 4N. We know this is wrong because the XL first of all cannot have been B2N, zygote is not triploid either. Point B. Pollen grain is N, XL is N, both these are haploid, zygote is 2N, that is also correct, and PEN, that is primary endosperm nucleus, is 3N. So without going through the other two options, we very clearly know the answer is going to be B over here. The next question. Pollination is a way to bring together the male and the female gametes since these gametes are dash A. Non-motile. If you consider the male gamete is in a way non-motile, okay, the female gamete for sure is non-motile. B. Release at the same time. Now we know they're not always, or rather the egg cell is never released, so we can't even consider that. C. Unable to cross-pollinate, but we don't always require cross-pollination, right? Okay. You also require pollination for self-pollination, okay, self-transfer. Cleistogamous. Now in cleistogamy you do not require pollinating agent as we have already studied. So the answer here is option A. Since both of them are non-motile, it is essential that the male gametes are transferred by the process of pollination, whether it be self or whether it be cross. Question number 5. Double fertilization in angiosperms is of two types of fusion between. So what does double fertilization contain? Option A. Male gamete and synergid cell. Male gamete and polar nuclei. Now we know there is no fusion with the synergid, so that's out of the question. B. Male gamete and egg cell, male gamete and polar nuclei. Now we know that this is correct. Let's go through the other options. Male gamete and egg cell, male gamete and antipodal cells. Now we have already understood or studied that the antipodals do not have a function as such. Male gamete and egg cell, male gamete and synergid. So this is again out of the question. There is no fusion with the synergid. So the answer here is option B. So I invite you students to go through all the various material in EduRev Infinity. You can go through the videos, the test papers, and a whole lot of material that is available. All of this being provided and prepared by NEET experts. And I'm sure you will increase your nationwide ranking by doing so. Thank you, students.